Hi,
Thank you very much, Zhanar, for agreeing to give a Build a Cell seminar, and you can get started.
thank you, Kate. Hi, everybody. My name is Zhanar Abil. I'm from the University of Florida. Excited to be here. Thank you so much for giving me this opportunity. I joined the UF last August, so I'm still setting up my lab. So most of the things that you will hear today will be from my work that I did as a postdoc in the lab of Christophe van Allan in TU Delft. And also I will share a little bit about my plans for research directions in an independent lab. So let's get started. I would like to start with the acknowledgements in case we don't have enough time in the end. I would like to thank Christophe van Allan for being an amazing advisor, for all the support and help and encouragement and expertise and kindness. It was great to be in his lab. And I would also like to thank the entire lab, especially Anna Restrepo, who is a PhD student that I trained. She has continued the work after I left, and now she is graduating in a few months, spreading her wings. She was an amazing companion, and a lot of the work that you will see today was done by her. And I'll also like to thank Women Zhao and Andy Allecton, my previous advisors, without whom I wouldn't be here where I am today. I would like to thank our colleagues at the Autonomous University of Madrid for on multiple occasions helping us with the purification of the proteins and with the discussions and with some of the experiments that they have, and our collaborators at the ESPCI Paris, Yannick and Thibault, for their discussions and with the help with analysis of with some of the data. And I would like to thank my current lab and current colleagues and collaborators for their support and help. And finally, I would like to thank the leading fellowship for the financial support when I was in Delft. So a few of the questions that I'm interested in is what sets life apart from non-life, how to build it, how to understand its origins. The question of so we biologists, we have discovered so many intricate underpinnings and mechanisms of how life works. However, it is surprising, or maybe not surprising, that we still do not have a unified theory of life, a theory that would explain how life originated here on Earth, or explain or predict or help us find life out there in the universe, or help us build synthetic life from scratch. And it is believed that bottom-up synthetic biology, or building a synthetic living system, or realizing synthetic life, would help us in developing this unified theory of life, and also help us in making progress in astrobiology and the origins of life. And also, I don't have to convince this audience that there was a lot of usefulness of bottom-up synthetic biology and the synthetic cell field in developing new and improved biotechnological and biomedical applications, such as drug delivery, drug design, developing disease models, biosensing, and biomanufacturing. When it comes to the question of how to build a synthetic living system, or a minimal living system, most of the research that has already been done can be categorized in one of these three broad directions. And one of them is the rational design and the manufacturing of the synthetic genome. Another direction is the rational design and the manufacturing of self-gene expression systems. And another broad category is the rational design of functional modules, such as replication or energy regeneration or cell division or compartment biosynthesis or cell signaling. However, when it comes to the question of what is life and how we can realize a synthetic life from scratch in the lab, all these directions are very useful, but they might not be enough. And here is why. One of the reasons that rational engineering might not be enough for the realization of synthetic life is that we currently do not have enough knowledge for designing a truly minimal synthetic genome. And besides, life is more than its genome and more than its DNA. Another reason is that life is not a sum of its parts, or life is not material. It's not, we like this metaphor of life being as a machine that you can put together from parts. It's a useful metaphor, but when it comes to realization of synthetic life from scratch, I think we can come up with a better metaphor. Let me bring up a metaphor of a river. River is not material. So if you take a bucket from the river, that water in the bucket is no longer river. It's matter, but it's not the river. The river is a process, so you have to describe it in the dimension of time. And so are so many other processes, like, let's say, a wave or fire or tornado or life. Life is a process in terms that it is made of multiple macromolecular components which are constantly in the state of self-regeneration. You take out a DNA out of a cell, and that's no longer life. It's just a chemical. All the macromolecules that the cells are made of, let's say RNA, gets turned over every few minutes or even seconds. Proteins get turned over every few hours on average. So you have to describe life as a process, and you have to describe it in the dimension of time. Therefore, it is not so straightforward to simply build it as one would build a car or a clock. Another thing I would like to address is the fact that we like to talk about life in terms of information flow, which is not wrong, but I think we're a little bit too centered on this view. When we describe the central dogma as life, as the transfer of information from DNA to RNA to protein, we also forget that a central dogma is not just about information flow, it's also about chemistry and catalysis. So if we represent the processes in the central dogma as a network of catalysis, catalytic reactions, then we would actually see that it's no longer a linear diagram, but a network or a cycle of chemical reactions, where two classes of macromolecules, proteins and nucleic acids, are in a constant process of cross-catalytic synthesis of each other from their substrates. And it is actually very important to also note that it's not just the protein. So what I mean by that is that, let's say proteins like RNA polymerase catalyze the formation of all the RNA in the cell, and RNA such as the ribozyme in the structure of a ribosome catalyzing the formation of all the proteins in the cell, including the RNA polymerases. So this constant cross-catalysis, which enables the system to be far from a thermodynamic equilibrium, is what we consider a living process. And it's not just transcription and translation, it's also decoding of the information. So yes, there is the information component where amino acid tRNA synthetases actually decode the RNA, the amino acid, the RNA into the sequence of amino acids. And also there is a component of energy regeneration, where energy from either light or food must be transformed and fuel this synthesis of all of these macromolecules. So all of these reactions together, including the reaction of metabolites and other pathways, constitutes life as this ensemble autocatalytic process. How does one recapitulate this kind of process in a test tube from non-living components? That is the question. And so what I propose is to use two processes by which life is usually described. One is self-regeneration, and the other one is evolution. To use these two processes to enable, to not only mimic a living process, but to enable its emergence from a less complex systems. So as I just mentioned, one of them would be self-regeneration. That would allow this network of reactions to be far away from a thermodynamic equilibrium. And the other one would be evolution, which is a driving force, as we know, that enables the persistence of the living process for at least 3.5 billion years. And we also believe currently that some form of chemical evolution also enabled the emergence of life in the first place. So I think by enabling these two processes, integrating them would help us in the realization of synthetic life from something less complex like chemistry. And this brings us to the two projects that I did in the lab of Christoph. One of them is in vitro evolution of a self-replicating DNA. And the second project that I'll talk about is in vitro clonal amplification of genes for improved in vitro evolution of proteins. So the first part of my talk is about how we can use self-replication as a seed module for enabling the evolution of a more and more complex self-replicating living systems. So if we want a system that is capable of evolution, we would need to start with a system that is capable of replication. And one of the simpler examples of that would be a DNA molecule that encodes for, let's say, a DNA polymerase using a self-free gene expression system. And then this expressed DNA polymerase would then amplify the template DNA from which it was expressed. This would make a positive feedback loop where at least part of the catalysts that are present in this system, which is a DNA polymerase, and DNA can also consider be as a catalyst, as synthesizing, regenerating parts of their components. And also, this is a system that would be capable of an autocatalytic behavior and Darwinian evolution. And I will explain later how that would happen. So it's in principle possible to have this system to evolve, but actually, can we do this in the lab? That was our first question. And another question was, can we not only evolve it, but observe adaptive evolution, where we can observe improved fitness? And by fitness, we mean the ability of this DNA replicator to improve its self-amplification. So these are the two questions that we were interested in. And as I just mentioned, we would need at least two systems. One of them is a self-free gene expression, and for this audience, I don't have to explain much about it. Will suffice to say that the self-free gene expression system that we use is a PureFrex, where all the proteins are purified to homogeneity and then put back into very defined mediums to enable self-free gene expression. And the other component would be a minimal DNA replication. And the machinery that we borrowed is from Basedo's Subtilis phage 529. It's a very simple system, and it's made of a DNA polymerase, a terminal protein, and a single-stranded DNA binding protein. So a DNA polymerase is obviously the enzyme that catalyzes the phosphodiester bond formation for synthesizing the DNA strand. The terminal protein is the initial substrate to which the subsequent nucleotides are ligated by DNA polymerase. And the single-stranded DNA binding protein coats the replaced DNA strand and prevents strand switching. So it's a very simple system that we can express in the pure system. So to have this self-replicating machinery, we combine these two systems, the pure system and the DNA replication machinery, into what you see here. The DNA polymerase and the terminal protein are encoded on a DNA template. So these are two individual transcriptional units that encode for these two proteins. And then this linear DNA construct is flanked by two origins of replication from phage 529. The DNA polymerase and terminal protein, once expressed by the pure system, would then make a complex where they would recognize the origins of replication and replicate the DNA from both ends, thus allowing the synthesis of two new DNA strands. And as a result, we end up with positive feedback loop where the more DNA you have as a template in the reaction system, the more protein is synthesized, and the more protein is made, the more DNA is made. So this becomes an autocatalytic, self-amplifying, exponentially growing system. We showed previously that it is possible to not only express the proteins from this DNA construct, but also to make it self-amplify in the pure system. As a result, you see here this band of DNA of 3.2 kb length, which is the size of our minimal synthetic genome. Now, is it possible to evolve this system? And to be able to evolve, we would have to start with a genetic variation. And the genetic variation we allow to passively accumulate by this innate mutagenesis of 529 DNA polymerase. So if you have some genetic variation in your population of DNA and you encapsulate it in phospholipid vesicles, and the reason why we need to encapsulate in phospholipid vesicles is to enable a genotype to phenotype link. So that the proteins that are expressed do not diffuse away from the DNA that they originated from. Now that you have a genetic variation and no more than a single DNA molecule is present in the vesicle, this allows differential amplification of certain variants. So let's say we have a beneficial mutation on one variant, it will produce more DNA. There is a harmful, a deleterious mutation in another variant, there will be no amplification. As a result, when you extract the DNA amplified by PCR and characterize it, we should be, in principle, be able to see enrichment of certain variants over others. And if we repeat this process multiple times, so we would recompartmentalize this DNA in fresh pure system and fresh vesicles, and repeat it multiple times, in principle, we should be able to observe enrichment of certain variants and, as a result, Darwinian evolution of a DNA replicator. To make these vesicles, we use a film hydration method. Oh, what happened here? We would start by solubilizing the phospholipids in an organic solvent and drying the solvent away so that the membranes are coating, the dry membranes are coating now glass beads. If we now rehydrate the system, the glass beads in our pure system, together with the DNA solution, this rehydrates the films into multilamellar vesicles. And then we would go on to subject these vesicles to multiple cycles of freezing and thawing, which would result in a higher fraction of these vesicles being unilamellar. However, most of these vesicles will still be polydispersed, meaning they will be varying in size. And usually what we get is giant unilamellar vesicles with an average diameter of 2 to 4 micron. Tess Beavers is an undergraduate student that I trained who did this experiment. She showed that if we stain the vesicles with a membrane, with a red membrane dye, and stain the DNA with a green intercalating dye, we would be able to see self-amplification of DNA in the pure system in vesicles, as is manifested in these bright green spots inside of phospholipid vesicles. We also show that it's possible to see it on a flow cytometer. If we stain the vesicles the same way, we would see a population of vesicles which are brighter on the green fluorescence, indicating that there's a population of vesicles with highly amplified DNA inside of them. So I just show you that it is possible to self-amplify the DNA inside vesicles. And the next, we wanted to see if it's possible to modulate the rate of evolution by using a variant of a DNA polymerase that is more error-prone. So we used a mutation in the DNA polymerase in the exonuclease domain in the position of 62. So it was a phenylalanine to tyrosine mutation that previously in the literature showed makes the DNA polymerase deficient in being able to excise the wrongly introduced nucleotide base. And we showed that in our system, when it's expressed in a bulk reaction, the yield of self-DNA amplification using this mutated variant is indistinguishable from that of wild type. And this is, again, in accordance with the literature. This mutation does not affect much its amplification, its replication activity. And finally, we wanted to see if evolution is possible with these DNA replicators inside of vesicles. So we used either the wild type DNA construct, which we would start with the original wild type DNA polymerase, or we would start with the mutator, the potential mutator variant. And we ran 3 independent experiments, 2 of them with the wild type and 1 with the mutator variant, and repeated these reactions of in-vesicle replication multiple times. In each round of evolution, we would measure the DNA concentration in the beginning and in the end of the reaction, which is 16 hours, by quantitative PCR. And as a result, you see this zigzag pattern of amplification and subsequent dilution of DNA. Here on the y-axis, you can see the overall amplification of the DNA in vesicles. So it's a ratio of DNA concentration in the end of the reaction to the beginning. And here we compile the data from all 3 independent evolutionary experiments. And as you can see, within 10 rounds of evolution, we could observe improvement in self-amplification in some of the evolutionary campaigns. The black line represents the wild type, and the green and the red one represent the 2 evolutions where we started with the wild type template. So in one of the wild type experiments, the red one here, you can see there was improvement in fitness in self-amplification from about 10-fold to around 100-fold. The green one did not improve in its fitness, and the mutator also improved the fitness really fast, after which we decided to increase the selection pressure by shortening the reaction time. So as a result, you see less amplification later on. Here we show that the round 11 of the wild type evolution, is actually improved compared to round 0 of the same wild-type evolution in a bulk self-amplification experiment. Now we wanted to see if this improvement is due to any specific mutations, if there was adaptive evolution happening, or are there any other forces that resulted in this improvement. So we deep sequenced every other round of all these evolutionary experiments, and we found that on average there was accumulation of around 5 mutations per DNA molecule by round 10 in about every independent evolutionary experiment. So we would start with 0 and would gradually increase the number of mutations per molecule. We also counted the total number of mutations found in the entire population of each evolutionary campaign. We considered as a mutation as every substitution that we could quantify as as a frequency higher than 1 in 1,000. And we counted hundreds of these mutations in each of these evolutionary experiments. I did not show it here at round 0. Before the evolution start, we could find maybe 2 or 3 mutations that are above this threshold of 1 in 1,000. So here we can conclude that there was a substantive amount of genetic variation produced by the system. But however, can we see enrichment of mutations and convergence of mutations? So I quantified the mutations that were enriched to at least 5% at any round of evolution. And we found at least a couple of dozen of mutation in each independent evolution. Many of them were silent and the others were not synonymous. And it is of note that most of the mutations were different from each other in 3 experiments. Even the second evolutionary experiment that did not show increase in fitness had very high enrichment for a number of mutations. The mutator version also evolved. However, as you can see, it's suspiciously synchronous enrichment of a number of mutations, and we later studied it in more depth and concluded that there was a contaminant from the evolution of the wild-type experiment that ended up amplifying in this mutator lineage. And as a result, we lost the original mutator genotype, and we got overcrowded with molecules from the other evolution. So unfortunately, we could not conclude how this mutator phenotype affects the rate of evolution. However, it is noteworthy to say that then the mutations that got finally enriched in this mutator evolution campaign were different in the end. So the exact mutationary profile was different from the other evolutionary campaigns. We went on to characterize these mutations to see if any of them confer beneficiary advantage to self-amplification. So Anna recloned some of these mutations, especially S79G and A80T, which are the mutations that are located in the exonuclease domain of the DNA polymerase. And we wanted to see if they improve the self-amplification of the DNA. And indeed, we saw a modest improvement of self-amplification in the bulk reaction. We also showed that these different mutations were located throughout the DNA polymerase and terminal protein, and that they also are not the same in 2 different evolutionary experiments. We also wanted to modify our evolutionary experiment. So instead of extracting the DNA from the vesicle every time we finish with one reaction and amplifying and re-encapsulating them in fresh vesicles, we wanted to streamline this evolution protocol to make it easier and faster to do. And so instead of extracting the DNA, we wanted to directly redistribute the DNA from old vesicles to freshly prepared vesicles and freshly made pure system. Using a freeze thawing methodology, which was previously developed in the YOMO lab, we wanted to see if it's possible to use this freeze thawing method to not only to redistribute the DNA between new vesicles, but also can we use this method to evolve the DNA. So Andreas Stan, who joined the lab late after I left, performed this, what we call continuous evolution mode of self-replicating DNA. And she showed that if we perform this self-amplification reaction in vesicles and then dilute them, let's say 1 to 10 in fresh vesicles, and then allow the reaction to proceed again, and we repeat this process multiple times, what she observed is that in the beginning, this was a kind of a steady state amplification and dilution where these rates were almost the same. But later on, the amplification was higher than the dilution rate. So hinting at the possibility that there might be some kind of adaptive evolution happening in this experiment too. We also deep sequenced this evolution, and we found that indeed there is enrichment of a number of mutations in this DNA population as well. However, we did not see as many mutations enriched as in the previous, what we call intermittent evolutionary experiment. We found that indeed there was not as many genetic diversity generated during this evolution mode. As you can see, this is the line for the continuous evolution, and it's already plateauing its genetic diversity generation at around 5. And we never reached hundreds of mutations per population. It was more of single or double digits of mutations that were observed at any round of evolution. So to conclude here, when comparing the two evolutionary protocols, the continuous evolution mode is easier and faster. However, the intermittent evolutionary mode produced a high genetic diversity, probably to this additional PCR amplification step, which resulted in higher mutagenesis rate. And this concludes my first part. I hope I convinced you that it is possible to evolve a self-replicating DNA in vesicles. And not just that, we can see adaptive evolution of linear self-replicating DNA. Now on to the second part of the talk. I want to talk about how to improve an in vitro evolution of proteins. So when we evolve proteins by directed evolution, we usually use some kind of host organism, such as E. coli or yeast, to express the protein of interest because it's easier and more robust. However, in some cases, it is more advantageous to use an in vitro expression system when the protein of interest is toxic or the conditions are not permissive to growth, or if we need to use substrates that are impermeable to cell membrane. So in these cases, it would be better to use an in vitro evolution. It would also be, if we want to evolve something in vitro, we would also be thinking of in vitro evolution of proteins in an in vitro system. However, we run into a problem when we want to do this, and that is when we express a protein in a cell-free system, we usually use a very high DNA concentration, template DNA concentration. However, if we want to link robustly a genotype to a phenotype, we need to be able to express the proteins even at very low DNA concentrations, a DNA concentration where there would be no more than a single DNA molecule per vesicle. At such low DNA concentrations, there is unfortunately very little protein expressed usually in cell-free gene expression systems. As you can see here, when we express YFP at very low DNA concentration, that would permit lambda not larger than 1, it's practically very difficult to evolve in such a setting. So then the question is, can we at the same time amplify and express the DNA in these vesicles? And this has been proposed and done in a number of labs, including Andrew Griffiths and the labs of Hohlfelder, where they use microfluidics or a bead display to sequentially amplify the DNA and then express the genes from that amplified DNA. What we propose is to simplify this protocol by a method that we call CAGE, or clonal amplification enhanced gene expression. In such a system, amplification and gene expression would happen in situ in the same pot. Instead of amplifying self, so amplifying a DNA polymerase, the DNA polymerase and terminal protein would amplify a gene of interest, which is flanked by origins of replication. And DNA polymerase can be added as a purified protein. In this case, we could observe, hopefully, amplification of and expression of the gene. If, on the other hand, it's not possible to find purified DNA polymerase or terminal protein, we could also envision a different approach where, a similar approach where the DNA polymerase and terminal protein are expressed, co-expressed with the gene of interest. But in the same scenario, amplification and expression would be happening in the same pot and at the same time. So we wanted to see that our vision is possible to achieve. So we first wanted to see if we can amplify the DNA in the pure system. And here you can see we quantify amplification kinetics in using quantitative PCR. And indeed, it is possible to amplify DNA at least a few orders of magnitude. Then we wanted to see if it's possible to express the protein in the presence of the DNA amplification machinery. And using LC-MS, we show that it is possible to also express the protein and to accumulate it to several orders of magnitude. And finally, we show that by monitoring the fluorescence of expressed YFP, that there's an advantage to amplifying the template of YFP comparing to using a non-amplified template, which is green here. Okay, we show that it is possible to combine replication and amplification, but can we do this inside of vesicles? Here we show combined amplification and expression, either using XP-CAGE, where we use expressed DNA polymerase, or pure CAGE, where we use purified DNA polymerase in vesicles. And we showed by qPCR that it's possible to amplify the DNA inside of vesicles. And we finally showed that it's also possible to improve the phenotype upon clonal amplification of DNA inside of vesicles. We measured this fluorescence of vesicles by flow cytometry. And here you can see a histogram of vesicles that express YFP only in the presence of pure. So there is no DNA amplification at a very low DNA template concentration. And these two histograms show the effect of amplification on the expression of YFP in vesicles. So as you can see, there is a higher dynamic range, there's much greater fluorescence in many of the vesicles. So here we show that it is also possible to improve the phenotype by clonally amplifying the DNA in vesicles. And finally, we show that it's possible to improve the sorting by doing a mock enrichment experiment. So here we mixed the gene of interest, so a YFP, with a gene that is unrelated, MIND, in a ratio of 1:10, and we wanted to screen the YFP using fluorescence activated cell sorting, and we wanted to compare a situation where there is no amplification to XP cage or pure cage. So this is a result of sorting, a single round of sorting when there was no amplification. And over here, you can see a result of sorting after a single round of amplification when it was clonally amplified. The amplification resulted in much increased brightness of the vesicles, so it allowed us to apply a more stringent gate. So we could use a much higher gate here. So we could use gate 1 or gate 2 to sort in clonally amplified samples. And as you can see, when we apply a higher stringency gate, we could result in a higher enrichment efficiency compared to no amplification sample. And another advantage of using clonal amplification is not only improvement of the phenotype, but improvement in recovery of the DNA from the vesicles. So when there is just the pure system, the amplification, the recovery of the DNA from vesicles is very difficult, whereas if we use clonal amplification, it's much easier to recover the DNA from sorted vesicles, giving us another benefit of using XP or pure cage. We did not perform an in vitro evolution experiment, but we did show you the results of mock enrichment, and hopefully this convinces you that this could be beneficial for in vitro evolution of proteins. And with that, I would like to conclude the second part of the results with the idea that combining replication and expression can be beneficial for in vitro evolution of certain proteins. And now I would like to discuss a little bit about the further directions in my lab, back to the questions of how to make a synthetic cell. So if we want to realize a self-regenerating, self-replicating system, which I introduced before, which could be regarded as an autocatalytic set, where all of the macromolecular components are regenerated within the system itself. Then we would be, it would be interesting to try to use what I call autogene networks. An autogene network is an extension of the DNA replicator that I previously introduced you to. So in autogene network, we will have another gene, let's say a gene of interest, the function of which would be coupled to expression or function of a DNA polymerase. And as a result, this coupling would make a synergistic gene network where the function of each component of the network would result in this holistic single fitness or self-amplification of the entire autogene network. For example, we could use a 16S ribosomal RNA, which is part of the small subunit of the ribosome. And Mike Jewett previously showed that it's possible to express 16S RNA and have it assembled into a small subunit of the ribosome. Then it's not a stretch to imagine such a ribosome expressing the DNA polymerase, and then this DNA polymerase would replicate the DNA, which would again make this positive feedback loop and exponential amplifying gene circuit. So in principle, any gene from any of the processes of the central dogma can be incorporated into an autogene network in a similar way, where all of the functions are put together into this synergistic, holistic network. And then the question is, can we add these kinds of functions and genes one by one and integrate them and evolve them in a cyclical manner so that finally we could arrive at a state which has a higher degree of self-regeneration and also has non-zero degree of fitness or self-amplification. And the closer we get to a state where it's capable of full self autocatalysis, then the closer do we get to the self into calling the system synthetic life. And hopefully with the many collaborations that we probably could foster, it would be possible to generate such to realize such a synthetic living system. To conclude, I hope I convinced you that it's possible to have a feasible approach into realizing synthetic life. And this kind of approach, which is based on self-regeneration and Darwinian evolution and system levels evolution, is a more holistic approach to bottom-up synthetic biology. And with that, I would like to thank you for your attention and to take any questions that you might have.
Thank you. Thank you very much. There are questions in chat. Can you see chat?
Yes, let me see. Okay. Bernard is saying, do you have to use pure for phytogenine reactions, or can you use TXTL? In principle, we could use TXTL, but the reason why we want to use the pure system for building a synthetic living system is because it's very defined, and we know exactly which components are in there so that we can modulate and replace some of them. However, when it comes to amplification and evolution of other proteins for in vitro evolution for whatever reason, in principle, we could use TXTL, given that it is not degraded by DNases and RNases that might be present in TXTL. If they are detrimental to self-amplification, maybe we can use inhibitors of DNases and RNases, and in principle, it's possible to use that. Christoph is asking, it may work in TXTL too, but it should be, oh, that's an answer, all right. And then Liam is saying, fascinating lecture. Have you checked how continuous and non-continuous reactions respond to changes in environment conditions, and such as changes in temperature? That would be a very interesting question to pursue. And we could study, yes, changes in temperature or any other ecological factors that we can think of, such as increasing the selection pressure, for example. Can we remove one of the components of the pure system gradually or abruptly? Yes, this would be a very interesting question to pursue, and how these two evolutionary experiments would perform would be very different. The intermittent evolutionary experiment would be safer because you extract the DNA and then we amplify it, and then we can re-encapsulate at a concentration that we want. Whereas in the continuous evolution, we have the risk of extinction. And this in itself is an interesting question, because then we can ask ourselves, well, is it going to go extinct, or can it go and undergo an evolutionary rescue? So that would be great to investigate. Peter is asking, saying, great talk. If 529 is rolling circle polymerase, how do you separate genomes into daughter cells? Do you cut the amplicon somehow? Yes, actually, the 529 and its amplification by rolling circle amplification has been done in the lab of Norika Ichihashi, who showed that it's possible to evolve such self-amplifying 529 by rolling circle amplification. However, yes, there is a problem, which is you making multiple repeats. So it will be a linear DNA with multiple repeats, and then to return to the original state, you would have to use a recombinase to circularize it again, which they did, and they evolved it in the presence of recombinase. However, the efficiency of the recombinase is not 100%, and there is still a lot of repetitive DNA even after recombination, which is why, even though it is possible to still do it in such a system, it might result in less efficient, in slower evolution. Whereas when it comes to linear DNA evolution, because we are regenerating the parental DNA every single time with a very high efficiency, we do not run into this problem as much. So that was a very good question. Hamid is asking, have you had issues with the trade-off between transcription and translation in the pure system? How have you addressed it for your current results, and how do you plan to approach the issue in the future? Yes, we did see some inhibition of gene expression due to the presence of DNA replication machinery, especially when we add double-stranded DNA binding protein, which enhances the amplification in the pure system. There is considerable inhibition. We addressed this by minimizing the concentration of the double-stranded DNA binding protein, and this improved the amplification. In the future, I think it would be beneficial to try other double-stranded DNA binding proteins and to see how else we can modulate the concentration, or maybe we could evolve this double-stranded DNA protein to interfere less with the gene expression. Seyun is saying, exciting work. Did you have a chance to look at your lipid vesicles via fluorescence microscopy? I am curious whether there would be some compartmentalization effects amplified by stochastic variance of kinetics of enzymes within the vesicles. Yes, we did look into lipid vesicles by fluorescence microscopy, and indeed there is some stochasticity in the system. There are many reasons for that. One of them is that the vesicles are of different size, and the crowding effect in each of those vesicles is probably different. How do we account for it? Well, currently, there's nothing much we can do about stochasticity if we use phospholipid vesicles. But if we use a microfluidic device to make a monodisperse vesicle population or a monodisperse emulsion droplets, then it is possible to mitigate this effect of stochasticity. And to some extent, this has been done in the lab of our collaborator, Yannick Randelis, and I think he's a better authority to answer that question when it comes to using the cage in microfluidics. Yes, Leah, you're welcome. GZ says, thanks for the brilliant talk. In the slide titled Repetitive DNA Self-Amplification, how can DNA concentrations reduce of each round after PCR? So if you mean the intermittent evolutionary mode, the DNA concentration is reduced simply because we extract the DNA, amplify, we quantify, and then we use exactly the amount of DNA that we need to bring it back to a low DNA concentration. And in the continuous evolution mode, we are monitoring the amplification by quantitative PCR, and based on those results, we would gauge how much dilution factor we would need to use. Max saying, do you know the mechanism why the DNA binding proteins are toxic for protein production? I'm not sure if DNA binding protein, so the double-strand DNA binding protein, um, the protein P6 that binds to the double-stranded DNA is actually known to inhibit gene expression in the phage 529, and it might be because it evolved to inhibit the expression of early genes in the phage 529. So that might be one reason. Another reason is simply because when we co-express the DNA polymerase and terminal protein, this co-expression also kind of interferes with expression. So therefore, using a purified protein may be advantageous to expressing it. Yes, burden could be the metabolic burden may be one of the reasons. Liam is asking another question. Did you compare different properties such as evolutionary rate to pace and pence? We actually did not compare. And I think this is a very interesting question. What are the different evolutionary rates, and in which case do we see I guess a higher selection efficiency, but yeah, we haven't done that. Yes, I think I answered most of the questions. Thank you, everybody, so much for your attention, for the great questions, and for having me here. I loved it. Thank you so much.
Thank you so much. Thanks for answering all the questions, and I'm really excited to see where that work is going.
Yes, thank you. Well, it was great. So, Kate, thank you for your for organization and for inviting. I love being here.
My pleasure. All right. Thanks. Thanks a lot. And thanks everyone. Have a great
Bye,
rest of your
everybody.
week.
Yes.
Bye.
Bye.